Good morning to one and all, and thank you so much for joining one day webinar. I just want to introduce Dr. Chittranjan Behera, who is our speaker for today's webinar. So, Dr. Chittranjan Behera is a dedicated and accomplished researcher affiliated with the CSIR, Council of Scientific and Industrial Research, from Indian Institute of Integrative Medicine, IIIM, Jammu. With a strong academic background and extensive research experience, Dr. Chittranjan has made significant contribution to the field of pharmaceutical sciences, particularly in the areas of oncology and drug delivery. Dr. Behera holds a PhD in pharmaceutical sciences, which he pursued from Guru Nanak Dev University in Amritsar, Punjab, and his doctoral research focused on exploring innovative drug combination strategies to enhance the effectiveness of anti-cancer molecules under the guidance of Dr. Prem Narayan Gupta at CSIR IIIM Jammu. He delved into the mechanism of cytotoxicity and sought to improve drug delivery system for enhanced therapeutic outcomes. Throughout his academic journey, Dr. Behera has demonstrated a high level of proficiency in various research techniques, including in vitro mammalian cell culture, molecular biology methods such as PCR and Western blotting, and microscopic techniques like confocal and immunofluorescence. He has also gained hand-on experience in animal studies, which includes the dosing of animals and evaluating the anti-cancer properties of test molecules. In addition to his research work, Dr. Behera has been recognized for his academic excellence, receiving prestigious fellowships and awards such as CSIR Research Fellowships and qualifying for GATE, and ICAR AICESRF PGS. His commitment to excellence is further reflected in his participation in various national and international conferences, seminars, and presentations, where he has shared his research findings and insights with the scientific communities. Dr. Behera's dedication to advancing the field of pharmaceutical sciences and his passion for innovative research make him a valuable asset to CSIR IIIM Jammu and the broader scientific community. His work contributes to the development of novel drug combinations and drug delivery systems, which have the potential to improve the treatment of cancer and other diseases, ultimately benefiting patients and healthcare worldwide. Dr. Behera is also a mentor in Drug Chemistry Research Center, DCRC. So now over to you, Dr. Behera, Dr. Chittranjan Behera. Hopefully the participants will gain more experience from your experience and by sharing your experience. Okay. So thank you so much. So now over to you. Sir, now I'm going to make you host so that you can present your slides.
Welcome to the one-day webinar on mechanistic investigation and synergistic interaction of drugs and their role in cancer treatment. So, as of already we know it about the cancer, it is the cancer represents the uncontrollable growth of cells in any organ or tissue of the body, and that can migrate to other parts of the body as well by a process called as metastatic status. And there are 6 intervening factors, the cancer cells that represents stimulate proliferative signaling, uncontrolled proliferative ability, insensitivity to growth suppressive signals, initiation of apoptosis process, and sustained angiogenesis, and finally tissue invasion and metastasis. And this is due to the exposure of carcinogen to the several environmental factors, lifestyles, diet, alcohol and exposure, and infection, stress, obesity, and physical inactivity. These are the contributing factors for initiating cancer, developing cancer in the human body. And it can be cancer can be a preventable disease that requires major lifestyle changes. So, as per the WHO report in 2020, cancer burden rises to 19.3 million new cases and 10 million cancer deaths. And as per the WHO report, common cancer in 2020, that is contributing to breast cancer and lung cancer equally around 2.2 million and cases, and cancer deaths in 2020 that represents lung cancer only the highest number, like 1.8 million cancer deaths. Then coming to the tumor microenvironment as a barrier to drug delivery into tumor tissues. So in this, the strong dense network that is composed of extracellular matrix and composed of collagen and hyaluron, and due to the increase in interstitial fluid pressure, the drugs cannot penetrate inside the tumor impervious to tumor microenvironment. That's why this is the TME modulation strategies. The dense tumor microenvironment can be modulated by the ECM degradation mechanisms, apoptosis inducing primary tumor priming, and vascular normalization strategies. So in this, I have done the tumor vascular normalization strategy I have opted in my PhD work, and thereby drug delivery inside the tumor tissue could be possible, and reduction of tumor masses. So in the tumor vascular system, that is the vascular system in tumor tissues is completely different from normal tissue, as it is heterogeneous in nature due to their organization and structure and functions. And the blood vessels are composed are very inconsistent in diameter, uneven shape, and abnormal bulges and blank ends. And the process of neovascularization, that is originating from pre-existing blood vessels to new blood vessels, that method is called as angiogenesis, which is possible due to because of the continuous release of vascular endothelial growth factor inside the tumor microenvironment. And that's why the vasculature is called as leaky vasculature in tumor inside the tumor microenvironment. For that, we have designed drug combination therapy, and there are some several advantages of using drug combination therapy, like that reduces the drug concentration specifically. First of all, again, the potential for drug-drug interaction it is increased, achieving the synergistic effect. And that synergistic effect is nothing but the interaction when two or more drugs interacts, and and that exerts a total effect of the drugs to be greater than or greater than to the sum of the individual effects of each drugs. Thereby, the enhancement of effect is seen, anti-cancer effect is seen, and that ultimately reduces the drug, reduces the dose-related toxicities and delays or decrease the drug resistance in achieving superior synergistic therapeutic efficacy. In that only maybe it involves two or more drugs in a chemotherapeutic regimen that reduces non-overlapping toxicity, also hinders the mitosis, reduces cancer stem cell population, and instigate apoptosis in cancer cell priming. So in that we have used micronutrients for this oncological intervention, like many micronutrients are there, like vitamin A, D, C, E, B6, B12, folate, zinc. These are the micronutrients: zinc, iron, copper, selenium are the vital micronutrients required for proper immune functions. But nowadays, various research have been performed using the selenium and the vitamin. So these are having some interesting antioxidative properties like vitamin C and vitamin E. And selenium also exhibits the cross-generating capability inside the tumor cells. And the immunomodulating and apoptosis inducing properties as are the very you can say that very poor anti-cancer efficacy they can exert, but whenever they can combine with another drug or some chemical molecule, they can synergize the anti-cancer effect in a fixed dose combination basically. So that is the first combination we have designed, that is using the D-alpha tocopherol succinate cross. That is a macronutrient with high moderate antioxidative activity and exhibits cytotoxic activity, induces apoptosis in various carcinomas, including breast, prostate, gastric, pancreatic, colorectal, as well as in neurons. And several combinations have already been previously studied using this combination with the apoptosis marketed drugs. So again, another adjuvant which we have used in studies in the calcium system. It is both both organic as well as inorganic form. That organic form is methyl salicylene, and inorganic form is sodium selenite. Both have been used in the used against the cancer cell lines to generate the anti-cancer effects. And In this case, we have used methyl cysteine just because it is also an anti-angiogenic agent that basically downregulates the key angiogenic regulators and results in muscular normalization and thereby decrease in the interstitial fluid pressure and leading to the higher intrathecal tumoral drug delivery. So in this study, we have used two different cell lines using this methylphenylcysteine with gemcitabine combination and alpha-tocopherol substitute with alkaline protein molecule, which is an in-house anti-cancer test molecule against pancreatic cancer cells like neoplastic cancer cells. So another studies like with MSC in combination with different anti-cancer drugs like tamoxifen, docetaxel, and paclitaxel. Some studies have already been performed in the several years. Apart from that, this is the drug discovery lead in the research work I have used the tripline protein. It was developed by Venkatesh Little, one of my uh, senior in 2015. And this molecule was not studied against different, in vivo models. And this it's very insoluble nature, and it is very uh, uh, exhibiting poor activity in solid tumor models. Hence, some strategies were required to improve this anti-cancer potential. That's why you have used the combination strategy to improve the anti-cancer potential of this molecule again with this combination of another adjuvant that is topical succinate. So it has screened against different cell lines like HL16, CD116, MGP with NPCP cell line. And impressive cytotoxicity was noticed in the neopathogen cell line. That's why we have conducted the studies this molecule against the in combination with alpha-tocopherol against neopathogen. So another drug that is the marketed drug that is gemcitabine as already been regarding this is marketed by marketed as Gemzar and approved by FDA, and it is being used in serious carcinoma and cell laboratories and metastatic breast and ovarian cancer. And this basically inhibits thymidylate synthetase, leading to the inhibition of DNA synthesis as well as cell death. And it is having several drawbacks like very short plasma half-life, like around 17 minutes. It's the rapid metabolism, cytidine DMAs, and low selectivity towards tumoricity towards the uptake and low availability. That necessity typical administration of gene leading to the increased systemic toxicity. And it is associated with several side effects like dose-limiting myelosuppression, such as anemia, neutropenia, and thrombocytopenia. Gemcitabine also elevate ALT, AST, and alkaline phosphatase levels. And it is several combinations also explored using the gemcitabine levels and several adjuvants as well. In past years, these are the examples and combinations of gemcitabine. So, and this is the first one, a combination of alpha-tocopherol succinate against in the combination with triple protein molecule. And the synergistic dose response was observed using the Confusion 1.0 software. That is its dose combination ratio 1 is to 0.1, which is the constant one ratio, and half of the activity dose was chosen for this combination to obtain the combination synergy, that is 16.99 of OS and 3.92 of that generated the synergistic response on combination with the indicative CI value 0.49, which is a synergistic value of 48 hours of time interval. Again, at 0.5, 0.75, and 0.9, this use of, attraction affected 0.5, 0.75, and 0.9 was also calculated using this confusion software. And again, it is also tested against the CAR cell line, which is the normal breast epithelial cell line. This combination is effective or it is toxic or not. To check out this phenomenon, we have done this experiment. And the synergistic dose 16.9 micromolar or 0.1 micromolar is found to be safe against this normal breast epithelial cell line. And as the dose increased, as the combination dose increased, and the cell inhibition was also increased. And the time dependent study was performed and found that the 48 hours is the optimum time period for this combination. Rather, there is no significant effect was seen in the 24-hour, and the effect was significant, similar, that is in the 72 hours, and there is no significant difference was observed between the 48 and 72 hours and 48 hours chosen for this combination selection with attain combination selection. And evaluating the nuclear morphology, it was done by normal bright field microscope as well as the scanning electron microscope, and the apoptotic body was seen in higher in number when the DOS and tripline protein was combined in comparison to the individual treatments. And similarly, the nuclear baby and the chromatin pigmentation was observed in the combination treated cells compared to the individual doses, and the positive control effect like for erasure. It was the number was counted using the uh, different fields, and it was counting by the different fields captured using this microscope. And the percentage cell apoptotic cell was calculated using the smaller number of apoptotic cells upon the total number of cells in the 100. And thereby it is justified that the combination of this molecule enhanced their apoptotic activities in the apoptotic cell lines and increased ROS generation and decreased loss generation and decrease in loss of mitochondrial membrane potential that was done by the uh, DCFDA dye and rhodamine 123 dye. And it was found at the wavelength of 485 nanometer and 530 nanometer. The loss generation was quite high in the combination treatment in comparison to the individual treatments alone here. And in the loss of mitochondrial membrane potential also, it was higher in the intensity. It was represented by this higher intensity of green fluorescence. It was indicated by the confocal microscopy at the 503 and 50 nanometers respectively. It is concluded that the higher, after combinating these drugs in a synergistic ratio, the ROS generation was significantly achieved and loss of mitochondrial potential also seen in this upper two cell line. So again, the in vitro cell migration is was performed. That is, this in vitro cell migration assays mimics the migration of cell in the in vivo models as well. So in that 1% FPS concentration, that is called a cell starvation method, was used. Whether to check out this phenomenon, it is assuring that the cell migration is not due to the cell multiplication, but only the cell migration is seen in this. So by using this wound healing, a formula was there, that is 100 minus area for 8 hours treated wound upon area of the 0 hour wound into 100. So for this 0 hour wound was captured, area was captured, and after that the 40 wound was captured, and the area percentage wound healing was calculated, how much the distance and the gap was generated between this cells. And apart from that, the combination indicates that is around 5 to 10% gap was gap closure was seen in comparison to the individual drugs coming with the natural, followed by the clonogenic experiment and the one with the autophagic experiment. So this is the clonogenic assay experiment in which the clonogenic ability of ability of the cells after combination treatment was diminished in comparison to the individual treatments, as well as the autophagy experiment was done using this acrylic orange staining. And in the combination treated cells, the number of autophagosomes like AVUs, acidic vesicular organs is seen in the combination treated cells in comparison to the individual treatment, and the higher fluorescence intensity was observed using the plate reader. ELISA plate reader was measured at the wavelength 526 nanometers. And this is the Western blotting experiment of Cascade 3 and PAR. Both have the intrinsic pathway, both these antibodies exhibit the intrinsic pathway of the process. And the Cascade 3 antibody and the PAR was observed in the near cell line, and the higher relative expression levels was higher in the combination treatment cells in comparison to the individual drugs as well as positive control for the cycle. This is the schematic representation of this combination with line protein and DOS in near cells, how they have entered into the cell and observe the mechanistic effect of this, like using, the ROS generated, and the ROS is increased, and a decrease in mitochondrial membrane potential, thereby increasing the sp3 expression as well as the PAR and cleared PAR, and that is finally the increased oxidative stress leads to the DNA damage and followed by increasing of the body formation. that is type 1 program is cell death, and initiated also the autophagy process, that is increasing the production of autophagy genes and the initiation of the autophagy process is now. Yeah, coming to another objective, that is to investigate another potential of gemcitabine in combination with the nuclear system. I've already told about the nuclear system. It's very compound and it is having anti-oxidative property. And it was screened with the gemcitabine drug, with the different cell lines like COP63, 5.1, PAM, MDA23, MDA23, and cells, with CF7 cells. Out of which was the individual effects like in A5 cells, it was around the lowest activity, I mean, lowest activity is 19 actually. And in the A5 the incident activity was around 1.08 really 1 microgram. That's why this acidity was chosen. And the one fourth acidity of this, both of these, like 22.5 and 0.25 micromolar were chosen after exploring different combinations against ratio-metric fixed dose combinations. We obtained this 22.5 and 0.25 micromolar synergistic ratio was for mechanistic studies. And after combination of this, we have seen that there was a six-fold dose reduction of gemcitabine was happened, and the further viability analysis pattern staining was done. And the synergistic combination of MSc and Jammu was also evaluated in the hypothetical that we normally risk typically whether the synergistic response is toxic or not, which is obviously. And the viability also was significantly lost in this 22.5 and 0.25 micromolar of methylphenylcysteine and gemcitabine. And followed by the staining analysis of all the synergistic doses, because we have found 4 to 5 synergistic doses, and out of these, which dose is appropriate for our cell line, I mean, for this study. To check out this phenomenon, we have done the nuclear morphometric analysis that is using staining, and we have treated with these 4 different synergistic ratios, and we found that 22.5 and 0.25 micromole is the best synergistic ratio, and the number of nuclei, number of nuclei and protein condensation is seen. Yes, that's why we have chosen 22.5 and 0.25 micromole. Our hallmark of apoptosis was seen in the cells as well. And the selected combination was those that fraction affected concentrated the mechanistic studies. Hence, rest of the mechanistic studies was done using this 22.5 and 0.25 micromolar ratios. And this is the dual dye staining experiment for the justifying the apoptosis phenomenon in this. We have done the acridine orange and ethylene bromide staining in A549 cells. The acridine orange staining reflects the 2 different signals, that is red and orange red and green signal. The viable cells and nuclei are represented by the green signals only. And ethylene bromide staining, and that only stained the dead cells. On the basis of that, we have counted using the Yokogawa cell cytometer, and that machine counted the nuclei, and as per that, the live early apoptotic and late apoptotic cell populations was counted. And it was found that the percentage of late apoptotic cell populations were significantly increased compared to the individual treatment. And in the untreated cells, there was maximum live cells were seen. That's why it was represented only the green color and no orange, red, or red color was found. And followed by the Ross generation experiment was performed using this the MSc and Jammu combination according to the as per the H2O2 standard. It was compared and found that this combination has the capability to generate a very good number of Ross inside the cancer cells, and in the in comparison to the normal as well as the individual blood proteins. Similarly, MMP also it was lost using this rhodamine 1,2,3 dystaining. And it was noticed that the significant MMP loss was happened in the after the combination treatment. In a similar way, this is the clonic forming experiment, clonic formation experiment. In that also this combination that leads to the decrease in the number of survival colonies in the epithelial cells in comparison to the individual treatment as well as the control having immune treatment. And this cell migration experiment was also performed using 1% FBS concentration that is called as self-starvation method. And it was the closure was calculated using software. And it was noticed that significantly the migration pattern was stopped in E5 to 9 cells in neutron, as well as the increase in the SNC in path expression in the A5 to 9 selector cells. Compared to the again this in vivo study was performed in this is the dosing setting, and in the day zero, we have inoculated the cells, that is EAC cells, and early carcinoma cells. And on the day 12, gemcitabine treatment was given to the group 3 and 4, and normal saline was given to this group 1. So gemcitabine treatment was given again in the 6th day and in the 9th day, and MSC was given on the 3rd day to the group 3 and group 4 as well. So this is the simultaneous combination MSC daily and gemcitabine on once every third day, and MSC and gem given every third day. So this is the simultaneous combination, and it is noticed that in both the group 2 and 3 and 4, the tumor size was significantly reduced in comparison to groups normal control and the gemcitabine alone. It was which was given in the group 2. So it was found that the improved efficacy was just because in the combination treatment group that might be attributed into the better availability of the tumor site inside the tumor tissue or site, as MSC causes tumor vascular normalization, as we have already read about this thing since the beginning. So in another set of experiment, we have performed this in vivo study again. That previous study was carried for this 13 days only, and this study was carried for the 22nd day. So it is, we have already known that MSC improves the tumor blood flow via vascular normalization. Therefore, another set of experiment was performed to check out this phenomenon. This is started on the day zero, inoculation of tumor was started, and after first, at the beginning of 4th day, it was treated with MSC, that is called as pre-treatment, up to 14th day. And after 15th day, gemcitabine was given to group 2 and group 3. And once every third day, it was repeated up to 21st days. And on 22nd day, the animals were sacrificed and photographed. And after that, on the day 13, since day 13, the evaluation of body weight was measured on once every third day. So there was no significant body weight was increased. It was nearly same to all the groups, including control. So in the group 1, that is given with the normal saline, one drug was given, and in 3 schedule was repeated as per the schedule. And the tumor volume was significantly decreased in the combination treatment with MSC and gemcitabine in comparison to the gemcitabine alone. Apart from that, the drug biodistribution study was also repeated to check out this phenomenon, like to calculate the drug concentration basically. So that's why we have used doxorubicin drug. And after 2 hours injection, the mice were sacrificed and the tumor mass was extracted, and it was homogenized to make a paste. And after using that paste, we have used in the HPLC analysis that we found that the drug concentration was increased in the group in those groups which we have treated with MSC in comparison to the groups treated with doxorubicin alone. And it was nearly found around this is around 0.5, and this is around 0.5, and after combination, the drug concentration was increased to 1 microgram per tissue, and that is around nearly the double of the amount it was increased. And that's why it is proved that MSC for this the tumor vascular normalization, and thereby the better availability of drug was uh, drug to the tumor tissues. And the histopathology experiment was also performed using this cutting the tissue section of those mice and tumor models. And after that, we have also seen in the presence of the complex microscope that that MSC causes tumor vascular normalization, and as doxorubicin having autofluorescence, that's why it was clearly visible that the increase in the red color intensity of doxorubicin indicated that due to MSC, doxorubicin was entirely reached to the tumor tissue in comparison to the doxorubicin alone, in comparison to those groups treated with doxorubicin alone. So this is the strategy we have done. And after entering into the plasma membrane, how it has instigated the apoptotic death, and and in the mice model, how it has enhances the tumor growth inhibition as well as better availability of the drug inside the tumor tissue. It was investigated in this movie. Thank you, finally.
Thank you, sir. And the presentation was very
Thank you.
informative. And thank you so much, sir. So I have two queries.
Yeah, sure.
So the first query is, what is Ross generation and how it is useful in cancer treatment?
Ross generation, that is, I have already explained, that is the reactive oxygen species generation. And as of already, we know that in cancer cells, the Ross generation is very obvious. As the cancer cell lines are residing in the humidified carbon dioxide incubator, where oxygen is not available. Only CO2 presence of carbon dioxide that the cells grows only. So in that, they are obviously they generate Ross reactive oxygen species in the cells in a very little amount. But we have used the H2O2 or vitamin C sometimes to generate the reactive oxygen species to compare with our drugs, whether the with our drug or combination they are generating loss or not. To check out this phenomenon, we have used the positive control H2O2, or either we can say that vitamin C or something like that. LPS sometimes people use to generate ROS. So to compare this phenomenon, we have the ROS generation is very efficient or not. To check out this, we have use this because in normally you can say in human body loss generation is very obvious. So after it is very necessary to scavenge those free radicals like H2O2 or peroxidases etc. that produces inside our body. So this is very useful in case of killing cancer cells instead of producing it inside the human body.
Okay, sir. Thank you so much, sir. Now the next question was, what is cell apoptosis and its role in the treatment of diseases?
Yeah, cell apoptosis is basically is a normal process that occurs inside the human body for the benefit, human benefit. Like that's why that is called as energy dependent mechanism of photolysis of cells, where ATP is being released. So that is there are two types of apoptosis, type one and type two. So by killing cancer cells, apoptosis phenomena is very necessary uh, as we induce apoptosis in the blood-treated cells, in cancer cells, or inside the in vivo models, that apoptosis phenomena is very much necessary.
Sir, I have one more query from the
Yeah,
participant,
yeah.
and the query is if we use two plant extract, okay,
Mhm, mhm.
and both individually both plant extract is showing synergy effects on cancer cell lines, but
Mhm.
when they are using a combination, in combination, the effects decreases. So
Yeah.
how can they present their data in a research article?
Okay. It is not necessary whatever the combination you are using, it always generates a logistic ratio. It may result antagonistic effect as well as the additive effect. So additive effect is also the CI values that comes nearly equal to one, or the antagonistic effect is very greater than one. So that comes in an infinite range ratio at the time. So that's why that combination will not be fruitful after combining those plant extracts using the software like calculation and computing or checkerboard method also. That's why in that case, that chemical molecule or plant extract, whatever you are using, that needs to be more refined or that needs to be, I can say, that needs to be changed. Or there are some rationals also, I forgot to tell you, that is the fixed dose combination rationals that always not work because the drugs in combination so that by different mechanisms. Either before combining the drugs, the mechanism of drugs should be known. Should be known that on which mechanism they are acting. That mechanism should be different. And the pharmacokinetic study also needs to be performed whether it is seen that the drugs must not be very wide. pharmacokinetics, and combination should not have overlapping toxicities. And yeah, that's it.
Okay. Now we have a query from Dr. Harshita Tiwari.
Yeah.
How do you see the future of this formulation? I think she is asking the question like you present your work,
Yeah.
so she just want to know how you will convert this research into formulation for marketed products or something else.
Yeah, we have, Dr. Chita, I would like to tell you that we have used that methylphenolphthalein and gemcitabine combination. And as if already I have explained that gemcitabine has several limitations like systemic toxicity, half-life, etc. And that's why we can modify this gemcitabine drug using the formulation approach. And as of in our study, I have not included the formulation approach using this gemcitabine drug. In another strategy, I have included it. But in this strategy, I have not included. That's why I could made a new formulation of gemcitabine, and it was combined. It could combine with the methylphenolphthalein system to achieve the synergistic response in human tumor xenografts as well. So that's why we had a drawback, and that we have used the plain combination. So everyone who was asked, like also in all the presentation, juries. So we can in near future it could be, I mean, like the main formulation would be better. Synergistic product. And methylphenolphthalein is completely safe. It is a cellular based derivative, so there is no need of making formulation for gemcitabine. I mean, methylphenolphthalein only for gemcitabine is safe. So that is a few drawbacks we have.
Yes, sir. Sir, thank you so much for your time. I think there is no query from the participants, and okay. So thank you so much, Dr. Chittranjan Behera, and all the very best for your future endeavor. And
Thank you.
hopefully we will again meet, and after this month, or at the end of this month, and we will organize once again one webinar.
Okay.
Okay, sir. So
Okay.
thank you so much for joining and providing your time, valuable time, and sharing your experience with Dr. Mystery Research Center. Thank you so much, sir.